bacterial endotoxin, host cell protein, protein content, purity, lipid content, carbohydrate content, and DNA content. After that, chemical treatment and diafiltration with chemical treatment, concentration, temperature, and time is monitored to ensure the efficiency of the process, and filtration, transmembrane pressure, permeate to ensure that the concentrate contains all the antigen. After complete purification of the bulk, now it comes to the sterile filtration of the bulk. And in the sterile, there is the pre-sterilization filtration bioburden, as we should ensure the bioburden level before implementing the sterile filtration. There is the filtration pressure to maintain the filtration pressure, the post-filtration integrity test to ensure that the filter remain integral till end of the filtration step. And we have the in-process testing, including the sterility, host cell protein, protein content, purity, antigen content, impurity content, and preservatives in multi-dose vials. After sterile filtration, it comes to the filling and stopping, where the common critical process parameter are the filling speed, cumulative process time, and cumulative process time out of the refrigerator, as the product should be maintained as in a cold storage conditions. So cumulative processing time out of the refrigerator is a critical process parameter. In this step, in-process control should include the fill volume, the headspace volume ratio, and those should be simulated in the media fill testing. After that, it goes for the lyophilization, where the shelf temperature is a critical process parameter to ensure homogeneity of the temperature, and chamber pressure, time, as well as the RAM time. And certainly at the end, we measure the moisture content. Capping, while we measure vial crimping value, sterility testing to ensure container closure integrity, visual inspection results to sort out any defective unit of the produced batch. This is an example. It's not an inclusive example. It gives an idea how to define what is critical parameter and how to define what is critical in process test to be implemented in every step. So any complex process should be divided into a serial of steps, and then in every step we should think what are the CPPs and what are the IPCs important to maintain the critical quality attributes. Another case study for bringing legacy product into compliance, and in this case, we started with a stage 3 when we have a capable process and we have a product performance that meets the predetermined specification with no cases of reported failures and no cases of complaints or incidents that those are significant. So how to bring this legacy product that is actually performing appropriately into compliance with the new approach of the process variation by implementing a gap assessment versus the requirement for stage 1 and stage 2. So from the process design, we should assess that the parameters assessed for their criticality and critical quality attributes has been defined, and variability of critical process parameters have been established, and acceptable range, those are resembling to the normal operating range, has been also defined. Quality risk management is implemented while establishing the control strategy, and risks are assessed and deemed adequate. Gaps of the starting material. So criticality assessment of the material should be implemented. Critical supplier qualification program should be in place and assess the supplier performance and compatibility of critical dispensed raw material with an intermediate container is defined. Hold times of the process intermediates those are critical for process intermediates, are supported with data, and the studies are there to ensure that these hold times are validated. Product contact material, criticality assessment carried out, critical suppliers are qualified, reports for assessment of material of direct product contact approved, like extractables, leachables, and these matter. The processing solutions like buffers, media, preparation, these preparations are assess the process of mixing, and mixing parameters are established, assess the homogeneity of the prepared solution are demonstrated, the filtration and bioburden control measures are established and well implemented, and monitoring for the bioburden in case of critical is established. Order of addition of material and the sequence of processing are well defined. Quantity of each component and associated range are established. Quality attributes of the solution, shelf life, expiration date of the prepared solution is qualified for critical processing solution, including testing aspects. For cell and virus banks, they are qualified. They are characterized by identity, purity, viability, stability is confirmed, and storage conditions are well defined. Viral clearance and inactivation activities studies have been carried out that demonstrate that the viral clearance are effective and we achieve complete inactivation of the virus. Impurity clearance if present, which include process product related impurities such as host cell DNA and the toxin, process additives such as antifoam, for example, antibiotics, all these are assessed and the clearance have been achieved with high capability. Filter validation. So whenever we will use filtration, whether diafiltration, tangential flow filtration, so the critical filters are assessed in terms of interaction with a process solution. Does it have a tendency for absorption? Is it compatible with the intermediates and the product? Is it appropriate from the capacity point of view? All these aspects should be assessed for every filter used. Chromatography resin and ultrafiltration membrane. So the qualification including carryover studies, storage, and confirmation at scale should be verified that it is available and in place. Sterilizing grade filter, bacterial retention study, integrity test parameters are in place. Integrity test is performed on the original solutions, so it is functioning properly. Media simulation for aseptic processing is there, completing for all aseptic operations with acceptable results. Bulk container integrity determined by risk assessment, qualified the integrity. Container closure system is qualified, ensured and verified that it is compatible and considered as a sterile barrier. And finally, transportation and qualification even is a part of the qualification and gap assessment for legacy process, because we should ensure that transportation of bulk solutions, intermediate or finished product are qualified and suitable for the product. To summarize, the new process validation approach and the enhanced knowledge management are mainly based on science and data, and they are linked to the risk. So all decisions concerning the control strategy, the detail, the number of PPQ batches, the decisions taken in accordance with the statistical analysis, those are based on data, based on science, and linked to the risk to the patient. From the regulatory expectations, we have highlighted that it is not a standard methodology, it's not a standard solution that is descriptive. that it's fit for the purpose is better than trying to do everything. So we focus on what is critical to the product, we focus on what is critical to the patient, and ensure that those are fully monitored with efficient manner. It is a continuous process. It's no more one-way process. So it built the quality from the design phase with proper understanding of the sources of variation and how to control them, because we know that repeat testing and inspection doesn't change result and doesn't build quality to the product. Implementing continuous process verification is a tool for continuous improvement and very beneficial in gaining more knowledge about the process and using this knowledge for a wise monitoring plan. And finally, in the monitoring plan, it should be based on the risk. It should avoid redundancy and avoid meaningless trend so that we maximize the value of our trending process. By that, I thank you very much, and I conclude my session, and I give the floor to David.
Thank you. Now we will launch the second poll. There we go. And give it a little bit of time. If you still don't see it, look for the poll function on your device and press on it to hopefully it will pop up. Again, 3 questions. Scroll through, answer the questions, answer the 3 questions is needed before you're able to submit your response. So we just give you a couple of minutes for everybody to quickly turn on their poll and submit their responses. Okay, we'll just give you 1 more minute for people to submit their responses before we go into Q&A. We have 1 question lined up, but that's just about all the time that we have for today. So let's end the poll now, and then we can go into the Q&A session. So we'll quickly show you the results of the poll. If you advance the slide. There we are. So Amani, if you can just spend a minute to explain why the answers are such in the poll questions.
Okay, thank you, David. The first question